Hi everyone, my name is Dr. Kellie Williford, and I'm excited to talk with you a little bit about my research looking at a specific population of brain cells and their role in stress processing, anxiety, and addiction. So, addiction is fundamentally a chronic cycling brain disease. And this cycle has three main phases. First, we call the binge or intoxication phase, and so that's just using the drug. This is followed by a withdrawal or negative affect. Affect is just another word for emotional states. These are the unpleasant physical and emotional effects after the drug wears off. And then this is often followed by a preoccupation or anticipation phase, in other words, craving. Now, stress is a major driver of this cycle, and stress can often cause craving and relapse. And individuals with addiction often report that this is one of the most common reasons for relapse occurring in the first place, is being exposed to stress. Now, a lot of different brain regions contribute to different parts of this cycle, and my work focuses in on the bed nucleus of the stria terminalis, or BNST. So that's right here. And it is really important for a lot of these negative anxiety feelings and fear and stress processing. And so if we look at, this is a picture of a mouse brain here. The BNST is located right in the middle. It's this small little triangular shaped brain region. And if we dive in a little closer, we see that there's actually a lot of different kinds of brain cells, again, these we call neurons, that do different things in this brain region. So some of the cells in this brain region can increase anxiety and increase the likelihood of relapse when they're activated, whereas other cells in this brain region decrease it. There have been a couple different medications that have been developed to try and target these different neurons, but they have not been successful at preventing relapse. So this tells us we really need a new target. Now, one group of neurons that have not been studied very much are called PKC delta neurons. That stands for protein kinase C delta. Don't need to worry about what that means for now, just know they're called the PKC delta neurons, and they really haven't been studied very much, even though they're actually a significant portion of the BNST overall. And so my first question was, could these be a new target? And what I wanted to ask was, what role do PKC delta neurons play in stress processing and anxiety? And so to start to get at that question, the first sub-question that I wanted to ask was, what does the activity of these neurons look like during exposure to stress? Are they even sensitive to stress? So how do we measure the activity of neurons? Well, what we can do is we can take a mouse and insert a fiber optic probe down into their brain. And this is actually the same kind of material that they use for Google Fiber Internet. And this fiber is able to detect light. And so then we can also put a molecule into the neurons that we're interested in. And this molecule actually lights up when these neurons become active. And so then with our fiber, we can tell whenever these neurons are active in a mouse as it's awake and moving around and doing things. Now, how do we stress mice? There's a lot of different ways that we can do this, and one task is called this novelty suppressed feeding test. And all that means is that we actually put the mice into this big new open box. Now, mice are creatures of prey. They don't really like to be in bright open spaces. It's not a very safe place for them to be, and they definitely don't like to eat out in bright open spaces. And so what we can do is food deprive the mice for one day so they're hungry, and then we put a food pellet into the center of the box. And we can see, okay, what do these neurons look like when the mice actually decide to approach and go ahead and eat this food pellet, which is a very stressful experience for them. And so what you're looking at here is this time zero is the moment when the mice decide to actually go ahead and eat that food. And then this is a measure of how activated the PKC delta neurons are. And we see that when the mice are overcoming this stress, these PKC delta neurons light up like crazy. They go, their activity increases a lot. And this is just quantified over here. We can see this is before they approach the food, and then this is when they approach the food, that activity increases. Now, another way that we can stress mice is a test called restraint stress. And so this involves just putting the mice into a tube and basically letting them sit there for an hour, which I don't know about you, but that sounds pretty stressful to me. And while they're in this tube, they go through these periods of time where they are actively wiggling their whole body and trying to escape. And what we can see is at the onset of when they are, actively coping and dealing with the stress and trying to escape, that's when we see these PKC delta neurons coming online. And again, that's quantified right here, where we see that when they start moving, that activity just ramps up. Now, a lot of other neurons in the BNST sort of get used to stress. So if you present the same stressor over and over and over again, then they actually stop responding. But what we see is if you stress these mice with this restraint stress for 5 days in a row, then they actually continue to respond every day. They're not getting used to this stress. They keep signaling that this stress is happening. And so maybe PKC delta neurons are different from other neurons in this brain region, and maybe they might be a better treatment target. So to summarize the last couple slides, we see that PKC delta neurons are indeed activated when animals and people, well, when definitely animals, we think people, are dealing with stress. We call this stress coping. So then the next question was, okay, if we artificially turn on these PKC delta neurons, if we activate them, what does this do to behavior? Because it could be the case that when mice are stressed, these PKC delta neurons are coming online to help them calm down. Or it could be that this activity of these neurons is actually causing a stressful feeling in the mice and making them feel more anxious. So we need to figure out which one this is. Well, how can we tell if a mouse is anxious? One test that we can use is called the elevated plus maze. And so, like I mentioned, mice don't like to be in bright open spaces, but they are really curious and they do like to explore. And so we can put them on this platform that has 2 arms that are enclosed and 2 arms that are open, and basically just ask how much time do they spend out on these open arms exploring, even though that's a place where they feel a little more anxious. And so mice that choose to not spend very much time on these platforms, out on these open arms, we can say that those mice are feeling more anxious. So if we activate PKC delta neurons, we see that they spend less time out on these open arms compared to when we're not activating PKC delta neurons. So again, this shows us that activating PKC delta neurons causes mice to feel more anxious. They're less willing to go out and explore. So it's an unpleasant feeling when mice have their PKC delta neurons activated. So then the next question is, how is this happening? What sort of information are these neurons even taking in to be able to cause these feelings of anxiety? What other brain regions are these cells listening to? So how do we look at what inputs come into any population of neurons? Well, what we can do is we can put in this molecule again into the brains of mice, and it'll start in the neurons that we're interested in, and then it will jump into all of the neurons that are connected. And so we can see this is a picture of a mouse brain here, and this is our starting population, and then all of the other white spots that you're seeing around here, this is just a zoomed in view, are neurons that are sending information to the PKC delta neurons. And what we can then do is look across the entire mouse brain. And just to orient you, this would be the front of it, and the back of the brain, the top of the brain's up here, the bottom of the brain is down here. And again, all of these white spots you're seeing around here are neurons that are sending information to the PKC delta neurons. So if we zoom in and look at what regions those are, we see there's a lot of different brain regions that are talking to the PKC delta neurons. And these brain regions process things like sensory input. So we have things like the auditory cortex responsible for hearing, and this post-piriform transition area responsible for our sense of smell and processing smells that are out in the environment. We also see a couple regions involved in what we call homeostasis. And so this is basically your body's way of keeping itself in balance and seeing, am I hungry? Am I thirsty? Am I hot or cold? So that you can then adjust and make sure that your body is staying in balance. And then lastly, we see a lot of regions involved in emotional processing. And so some of these regions, like the subiculum and the central amygdala, are really involved in processing a lot of negative emotions like fear and stress. And then some of these regions are responsible for some of the more positive feelings like reward and when things go wrong, things like addiction. So if we put all of this together, PKC delta neurons are really trying to figure out what do I need, so that your physical state, what's going on around me, again, that sensory input, your hearing, your smell, and then how do I feel about it? What's my emotional state? And so then the last question, and maybe the most important one, is what does this all mean for the mouse when PKC delta neurons become active? And so once again, we're going to turn back to that elevated plus maze. And this time we're just going to record the activity while the mice are exploring around. And what we see is that when mice return to these safe closed arms, we see that the PKC delta neurons are actually turning off. Their activity decreases when the mice feel safe. And then conversely, what this graph is showing you is along this axis here is how active the PKC delta neurons are. And then this is how long they spent in the open arm on that visit. So while the mice are around exploring, sometimes they'll be in the closed arms and they'll just poke their head out and then they'll turn right back around and come back into the closed arms. And sometimes they'll poke their head out and then go ahead and decide to explore. And what we see is that when the PKC delta neurons have a lot of activity, the mice immediately turn back around and go back into the closed arms, seeking safety. They immediately return to safety. But sometimes when the mice enter these open arms, there's not very much PKC delta activity, and that's when they decide to go ahead and stay out and explore. So if we put all of this together, what we really see is that PKC delta neurons are tuned to detect threats and stressors, and then integrate that with your internal state and figure out, okay, what should I do about this? If it signals that there's some sort of threat or stressor, then this would drive you to seek out safety. But if not, then it'll allow you to be a little bit more exploratory and try to figure out what's going on around you. Now, the question you may be asking is, how does all of this relate to addiction? Well, one thing I didn't mention is that, yes, we see that PKC delta neurons turn on in response to stress. We also see that the brain actually starts making more PKC delta after exposure to stress. And so in the case of addiction, it could be that these neurons are becoming overactive and that they are constantly signaling threat or stress, or that there's too much danger, even to things that aren't actually dangerous. And so then these stressors are causing you to say, okay, I need to return to safety. There's something out here, there's something going on, and I need to return to safety. Well, what is something that's safe in the context of addiction? Those are the previous coping strategies, things like the drug. And so what we think may be going on is that these PKC delta neurons become overactive and cause people to seek out what feels safe and return to their previous drug that they are addicted to. And so what we would like to do then is move on to say, okay, what happens if we block this overactivity? Can we actually interrupt this cycle of addiction and help people to remain abstinent so that they're not feeling overly stressed by these things in life that are stressful? Because we can't really avoid stress, but can we help prevent too much feelings of anxiety and this drive to cope with that in negative ways by blocking activity of PKC delta cells? And so this was a lot of the work that I did in my graduate time at Vanderbilt. And so work in that lab is continuing on trying to investigate these PKC delta neurons. And now I'm currently a postdoctoral fellow at Duke University, where I am actually moving on to a different phase of the cycle, trying to understand craving. We know that sometimes experiencing craving leads back into relapse and using that drug. But a lot of times, people are able to experience craving and then maintain that abstinence. And so my question now is, what goes right in the brain every time someone is able to actually maintain that abstinence, even in the face of craving? And can we amplify that to help people maintain abstinence in the face of craving, in the face of stress. So with that, I just want to thank my lab where I did most of this research. Like I said, this was at Vanderbilt in the Winder lab, a really amazing group of people there. And I'm currently at Duke University in the Collective for Psychiatric Neuroengineering, continuing my work on addiction. I also want to thank my family, who are a constant inspiration for me in continuing this work. And if anyone's interested, these are some of the papers where you can find more information about these PKC delta neurons and about addiction in general. If you have any questions about this, please feel free to reach out. I'm happy to answer any questions. So thanks so much, and enjoy the rest of your day.